educational video. So in this video I'm going to discuss about an introduction regarding with the microbiology. So let's see the definition of microbiology. Microbiology is a branch of biology which deals with the study of microorganisms, the organisms which cannot be visible with our naked eyes. They can only be visualized with the help of a microscope. Such types of organisms are referred as microorganisms. For example, fungus, virus, bacteria. So there are different kinds of fungi, different kinds of virus, different kinds of bacteria are there. So these are all referred as microorganisms because of their micro size. So next we'll see the father of microbiology. The father of microbiology is the Antony von Leeuwenhoek is a Dutch scientist. So he is known as the father of microbiology because of his great invention to the field of microbiology. So he's the very first person to invent a microscope. So he invented his own single-lensed microscope and also is the very first person to document a microscopic observations of the muscle fibers, microscopic observations of the bacteria, spermatozoa, red blood cells, crystals in gouty toffee, and blood flows in capillaries. So this picture shows Antony von Leeuwenhoek is a Dutch scientist. Next, we will see the father of modern microbiology. So the father of modern microbiology is Louis Pasteur. He is known as a father of modern microbiology because of its great invention to the field of microbiology after Antony von Leeuwenhoek. So he proposed a germ theory. So let me explain first what is a germ theory. For example, consider this is one container. So inside of this container there are food substances. So this container is exposed to the air and it is not closed. So now what happens, whatever the bacteria are present or whatever the microorganisms are present, for example this is a microorganism, so whatever the microorganisms are present in the environmental air, so they enters inside the container. So because the container is not closed and it is not sterilized, so it is unsterilized one. So if the container is not closed and it is unsterilized, so there may be there will be a development of microorganisms on the surface of the food because the food is got contaminated with the microorganisms. So for example, this is the container which is closed and which is sterilized. So the food substances present inside, they will not get contaminated because the container is closed, it is not opened. And hence he proposed that in sterile environment there is no development of microorganisms. In open and unsterile conditions there will be a development of microorganisms. And also is a very first person who introduced a concept of vaccination, microbial fermentation and pasteurization. First let me explain about the pasteurization. Pasteurization is nothing but exposing the food substances to a mild heat to eliminate the pathogens, and to extend the shelf life of the food. For example, this is the container which is heated. So to eliminate the microorganisms present inside, pasteurization temperature is 70 to 75 degrees Celsius. So this is the pasteurization temperature. So under this temperature no microbes can survive up to 15 seconds, or for the effectiveness we can do it for 1 hour. So if we expose 1 hour, it is very effective treatment to kill the microbes present in the food substances. Next is microbial fermentation. Is also very first person to document that microorganisms are responsible for the process of fermentation, and also he invented vaccine to anthrax and rabies. So he invented vaccines through anthrax and rabies. So this picture shows Louis Pasteur. He's a French biologist, microbiologist and chemist. Let's see the fields of microbiology. So there are two different fields, that is a basic field and applied field. Under basic fields there will be study of microorganisms. Whatever the microorganisms are there, they will be studied under this field. Then their process about microbial metabolism, about their physiology and disease related, about their immunology, etiology. And under the applied, so there is disease related field about their infection, what are the infection caused by the microorganisms, and environmentally related, so how the microbes are helpful to the environment. and industrial important, how the microbes are helpful to the industry. So next we will see the characteristic features of microorganisms. So we all know that microorganisms are small in size, so they're only visible with the help of a microscope. They have a very rapidly growing cells. For example, they have, this is a one cell which is rapidly divided into another two cell. Further it is divided into another two cells. So there is a rapid cell division in microbes. So this is one of the characteristic features of the microorganism, and they have a habitat, so they can survive in almost all kind of habitat in harsh environments. They can adjust to the any kind of environment. So they live in population, they not live in alone, so they live in population in group. So that group is nothing but is a biofilm, so which is attached to the surface, or there may be any solid surface, either it may be a dead tray or a pipe. So some microbes are harmful, some microbes are beneficial. So this picture clearly tells about the size of microorganisms. For example, atom which is zero point 0.1 nanometer. So protein is ranges from 1 nanometer to 10 nanometer, that is 9. So here flu virus which is 100 nanometer, and mitochondria that is 1 micrometer, and bacteria is 1 micrometer. Here plant and animal cells are about 50 micrometers. So is a Carl, who is the scientist who invented a three domain system, three domain of classifications of all type of organisms such as Arachea, bacteria and Eukarya. Let's see about domain Arachea. So this includes all the prokaryotes. So the microbes, whatever all the microorganisms comes under the Arachea, they are all prokaryotes, because their DNA is not enveloped with the any kind of membrane. It has a naked DNA. So there is no nuclear membrane. These are the oldest species. They have a distinct biochemistry. For example, methanogens and thermoacidophiles. So this is the typical picture, shows the prokaryotic cell, or this is one of the example for the bacterial cell also. So here we can see the DNA is naked, around which there is no membrane. So hence it is referred as a prokaryotic cell. Domain bacteria. This is also a prokaryotic. So it includes all bacteria which are prokaryotic. So the membrane is made up of diacylglycerol lipid. It has a ribosomal RNA. So of course no nuclear membrane is present. Includes all types of bacteria. Example is cyanobacteria, actinobacteria and gram-positive bacteria. Domain Eukarya. So it includes all the eukaryotes. For example, animals, plants and fungi. So this is the typical picture, shows the eukaryotic cell, which consists of a nuclear membrane. Here nucleus is surrounded by the nuclear membrane, hence it is the eukaryotic cell. Next we will see kingdom fungi. Kingdom fungi includes mushrooms, yeast, molds, mildew and rusts. So what are the characteristic features of kingdom fungi? So the most fungi are grow like tubular filament called hyphae. So they grow like a tubular filaments called hyphae. So hyphae are combined. So the intermass of the hyphae is called a mycelium. The walls of hyphae is strengthened with the protein called chitin, which produces the structural rigidity, for the hyphae. These are all the hyphae. These hyphae are combined together to form a mycelium. So these single segment separation is called a septum. So the membrane around the hyphae is called a cell wall. So there are 1 lakh species have been identified under kingdom fungi. So the cell wall components are chitin and N-acetyl glucosamine. So these are the structural components of the cell wall of the fungi. So the fungi disperse themselves by releasing spores. The spores are very hard to kill. For example, spores of feast rust fungus have been found at 4000 meter in the air. So more than 1450 kilometer from the place they were released. So no wonder that most fungi have worldwide distribution. So fungi are heterotrophic. They depend on another organism for nutrition. So some fungi are saprophytic. So they get the nutrition from the dead matter. They're saprophytes. So they get the nutrition from the decay matter or dead matter. Some lives in mutually beneficial symbiotic relationship. For example, rhizobium is a bacteria which helps to fix a nitrogen in leguminous plant. So thus helps the plant to fix the atmospheric nitrogen. Some are parasitic, which causes the severe infection in plants, animals and humans. So this is one of the example for saprophytic fungi, that is a mushroom which is growing on a dead matter. So this is also a mushroom, saprophytic fungi, which gets the nutrition from the dead matter or decaying matter. You can see in the picture which is growing on the dead tree because it's getting a nutrition from the dead tree or decaying tree. So this is the example for the mutual symbiotic fungi, that is rhizobium. So the rhizobium is present in the root nodules. So these are the nodules. So it is present inside the root nodules, helps to fix the nitrogen. So this is the microscopic view of the root nodules. You can identify the rhizobium present inside the nodules. These are the rhizobium bacteria. This is one of the example for the parasitic fungi, that is Ustilaga medis, which causes infection in corn. That infection is called corn smut. This is also one of the example for the parasitic fungi, that is Candida, which causes skin infection in humans. This is also one of the example for the parasitic fungi, that is a trichophyton. This is a microscopic view of the trichophyton, which causes a skin infection to humans. He is a scientist, R.H. Whittaker, who proposed a 5 kingdom of classification of organisms, that is Monera, Protista, fungi, plant and animal. In the next video, we will study in detail about the each kingdoms. I hope you understood and thank you for watching. Hi friends, subscribe to my YouTube channel and also press a bell icon to never miss an educational video.